afternoon all. On behalf of the Faculty of Pharmacy, Parul University, I, Dr. Bindya Johan from School of Pharmacy, welcome you all in today's seminar. For this webinar, we have a very special renowned personality with us, Dr. Sunil Rodamani. It's a pleasure for us to introduce Dr. Sunil Rodamani to all of you. Sir is currently working as a scientist one in the Dr. Prabhakar Kore Research Center. He is having the more than 11.5 years of the research and the training teaching experience. Sir has completed his bachelor in the chemistry botany zoology in the 2007, MSc in the biotechnology 2009. Sir has also done the MPhil in the amelioration of alloxan-induced diabetes mellitus in the Swiss albino mice, and Sir has done the PhD in studies on isolation and the characterization of bacteria producing bacteriocin in 2014. Sir has also qualified the GATE exam with a more than 88%, and Sir have been the guest lecturer and PhD fellows in the Karnataka University. He has worked as a teaching assistant in the Dhanvantri University and scientist one right now in the Kerala University. Sir has also been having the various hands-on experience in the GLP, in the gel documentation, AFM, FTIR, Raman spectroscopy, and also worked on the cell-based assays. And Sir has also received the various research fellowship by the URS, that is the University Research Scholarship from the Karnataka University, has also received the PhD fellow in the DBT IPLS by the DBT in 2013 and the 12, and Sir has also received the SRF from the University of Agricultural Science and the Dharwad. Sir is also currently working as a reviewer and editor in various journal like the Springer in the Bantam Science Ambassador in the Journal of Applied Pharmaceutical Science and the MPDI Journal Applied for the Reviewer of the Task. So it has also published various research publications which is having a cumulative impact factor is more than the 55, and also being has presented with various national and international conferences his paper. With this the small biodata, I welcome you sir in this webinar.
Yes, thank you so much for giving me an opportunity to just present a piece of data and giving me an opportunity to be along with the scientific community with Parul University. This is the first my experience with Parul University, and I hope it will be a nice exchange of knowledge. And to my topic, I think I am audible to you all.
Audible sir? Yes sir.
Yes. So today's topic of discussion is human gut microbiome, health, aging, and disease. So in the era of COVID-19, uh, the importance of health is very important. Keeping health in a good condition is a challenging task because uh, a person or a people experience variations in the environment, variations in the our nutrition, variation in the seasonal variation that we also encountering with the hurdles. So these all what will do, it will affect on health. In what way it will help? What way it will affect? So one thing is gut microbiome. So gut microbiome, how it is connected. Today's discussion is that what I am telling to you that or discussing with you is that human gut microbiome is very important in maintaining the health and disease. So in disease condition, what type of bacteria, what type of virus or fungus or actinomycetes or can call microbiota, okay, microbiota or microbiome is similar words, but ultimately we are addressing the live organisms other than whatever we consume synthetically. Okay, so our gut from alimentary canal, that's the alimentary canal from the mouth to the anus, so it contains the different type of biome called as microbiome. They are non-visible through naked eye. Microscopic studies are very important to study their effect and enumeration. But now, whatever the bacteria or the microbiome present in the gut that are classified under various groups, but uncultured bacteria are many, uncultured fungus are many, uncultured viruses are many. So how to identify those uncultured bacteria, viruses, or fungus. So that is one answer is NGS, next generation sequencing. So that is very important if you want to study the bacteria or microbiome in a huge set of data. So NGS is a platform where microarray based also. It is very high throughput than microarray. What happens during a PCR is that we identify one bacteria or two or three or four multiplexing. If you do also, you end up with identification of some few bacteria. But to identify a huge number of bacteria, we have to go through microarray. And if you are high throughput technology is at NGS, next generation sequencing, if you use that, we are able to identify various type of bacteria, cultured bacteria also, uncultured bacteria also, whether you need media for the growth or no media also. So in that case, NGS is very good studied in many aspects in health and disease, like in a cancer, diabetes, various type of malnutritions, uh, liver disorder, so kidney disorder. So everything like whatever disorder I'm telling you, these are always connected to one or the other way. It is connected to your bacterial population, fungal population, or virus population, and this biosis leads to severe conditions. How to mitigate that is very very important. We consume what curd we consume buttermilk. So what is different between curd and the buttermilk is the type of bacteria present in curd, and buttermilk is different. So that we need to understand. So if we understand that, I think uh, we are able to fulfill the need of the nutrition, and we can able to control aging also and disease. And nowadays people are feeling like they're obese in nature, and they eat many of the time bakery products. So the bakery products, what bakery products are, I only if you consume bakery products, there is increase in the weight, or there is like body like metabolism is very hindered. So, what is the implications of using bakery products or fast food? So that is very important to understand. And then if you see the current uh, arena for food system, and if you see the food system of our Indian from Kanyakumari to Kashmir, or from Rajasthan to West Bengal, if you see the food habits, it is very suitable to our tropical country, that is India. So if anything, if you are trying any other than the whatever is proposed by our ancestors, so there is a start of our new disease. So how to maintain it? It is our research team. So I am working with Shivani Tendulkar, a PhD student, and Ashwin Hattiwari, research center, Professor Abhijit Bhatkal, Satish Hegde. Now he shifted to Puducherry. Our head of department, or earlier the head department, Shivi Jalalpure, present head of department, Ramesh Paranjape. So recently he joined. First area is gut microbiome, cancer, chemo resistance, and biological chemistry. So this is my research team, small research team. My collaborators are same: Lokesh Shastri, Mahesh Chowdhury from Pune, NCMR, Sudhir Manwari from Karwar, Nagapati Adari from Gokak. So Sachin Aligar from CFTRI Mysore, Prakash Halami, principal scientist CFTRI Mysore. So he is also being in collaboration. I'm getting some inputs from Professor Halami for our projects work also, collaborators. So what is the human microbiome, gut microbiome? Human body hosts trillions of the microorganisms in different niches. Their genome is an integral part of our too. So hence the complete set of the microbial genes in our body is termed as microbiome. So you are seeing the oral microbiome, skin microbiome. So if you are the digestive tract microbiome, urogenital microbiome. So if any variation in the oral microbiome called as oral dysbiosis, any variation in the skin microbiome called as dysbiosis, and dysbiosis is what one type of population of the bacteria or the microbe is increasing, and another population of the bacteria or the microbe is decreasing. So if in case that happens, it's called a dysbiosis. So dysbiosis leads to improper balance of salts and acids in our body part, and that will lead to stress, that will lead to toxicity, that will lead to inflammation. So in this case, inflammation is a cause of the cancer. We are saying inflammation is cause of the many diseases. Okay, if you mitigate that by permutation combination or giving tablet in the form of you are seeing that lactic acid bacteria is one of the tablet given in the digestive tract infections whenever diarrhea or dysentery happens. So doctor is prescribing to lactic acid bacteria filled with tablet filled with lactic acid bacteria as a medicine. So what will it will go and do the work? It will revive the bacteria which are very very important for our gut or body system to function. So what it will exactly do and what are its importance in stomach? If you study that, I think the importance of microbiome we can able to understand. So microbiome lives and evolves in our body as we do, and now a very crucial part of our system. So it is present with us from the after the birth, from the birth, and colonize in our system. It will be remain with us till we take last breath, and after that also degradation also it is there. So it is a part of our life. And if you see the number of cells and the number of bacteria present in our body, the number of bacteria present in our body is a huge in number rather than the number of body cells in our system. So they play a very important role in our metabolism, immunomodulation. Immunomodulation is very important. Mood and behavior is important. So it will maintain that what microbiome it will maintain. So microbiome is unique to every person, much like our fingerprints. So if that is a signature of every human being, every individual, X person, Y person, or Z person, we can able to distinguish based on their signature or a fingerprints of microbiome present. So gut microbiome versus COVID-19. See here, we should connect COVID-19 and gut microbiota. If it is normal gut microbiota, if this biases happens, then what is going to happen? And if we, that will lead to like a immuno compromised patients. And if you see the person with a good immunity, what are difference between immunity and the immunocompromised patient is that, so difference in the type of microbiota they have in their body system. So gut microbiota diversity is decreased in the old age, and COVID-19 has been mainly fatal in elderly patients, which again points to the role of gut microbiota may play in the disease. So there is a recent study that if you have the good gut microbiota, you can able to fight with the invading bacteria, fungal or viruses. Invading, whatever may be, virus, fungus or bacteria. If it is coming into the body, you can able to fight with the incoming foreign particle, that is virus in the case of COVID-19. Improving gut microbiota profile by personalized nutrition. But personalized nutrition is one where you're going to consume high calorie, also high fiber content, high protein content, and the diet stuffed with uh, a particular type of beneficial bacteria, that it will suffice the need of microbiome. And supplementation known to improve personal nutrition supplementation known to improve immunity and can be one of the prophylactic ways by which the impact of this disease can be minimized in old people and immunocompromised patients. So just you remember, if you eat curd mixed with some sort of sugar, okay, how much of energy you feel, and when you eat other stuff of food, how much energy you feel. That difference is all about, it will tell about the importance of gut microbiota. And more trials may be initiated to see the effect of post supplementation of personalized medicine or personalized functional food, including prebiotic, probiotic, along with current therapies. So these are very important. Prebiotic are the food for the probiotics, if I'm not wrong. These are the food for the probiotic. Probiotic are the organisms which are beneficial. They are present in the stomach. They are present in the different body system, and mainly a beneficial role. They play a beneficial role. So we leave our mother's sterile womb and enter a world full of bugs. And before parturition, we are in our mother's womb, and as far as possible, it's very sterile from the other type of microbiota. But when you take birth, you the baby, the new infant starts to colonize its gut with a different type of bacteria. Okay, so that is very important to study here. Babies born by cesarean section have dramatically different gut bacteria to those normal delivery, according to the largest study in this type of field. Okay, Wellcome Trust, the Sanger Institute and University of Birmingham assessed how the microbiome form when we leave our mother's sterile womb and enter a world full of bugs. Full of bugs means that all type of microbiome, or all type of pathogenic bacteria. In the sense, bugs are very dangerous. So cesarean babies are more likely to have some health problems later in the life. So, probiotic food that can help to improve digestion and boost immunity. That we are here going through many type of food that I'm telling you. These foods are day-to-day food of Indian people. Yogurt or a curd, dahi is one of the best available and natural probiotic food that you can add to your daily diet. Kefir is a uh, good source of many nutrients. Powerful probiotic than yogurt has potent antibacterial properties, improve bone health, and lower risk of osteoporosis. So that is very important. You may be feeling that these all are very common, and we are discussing in the present lecture or present discussion. But we are forgotten these all, and we are switched on to funny. We switched on to uh, burger, pizza. So I'm not denying those are also good food, but our tradition is very important to maintain. Idli, driven by bacteria, they constitute total Lactobacillus mesenteroid family of bacteria, and genus Bacilla emerged as the most important organism in fermentation. So these are very important. Indian cheese, that is paneer, reduces the risk of breast cancer, makes teeth and bones strong, aids in weight loss, ensure healthy digestive system, great for those having diabetes. So that is very important paneer. So fermented soybean, that akhuni, protein-rich legume, uh, in otherwise traditionally protein sparse diet. Okay, these are used kimchi, one of the other food used in irritable bowel syndrome and colon inflammation. So these are the food of our system, and then how we will enrich the microbiome and keep the microbiome in a balanced system. That's very, very important. Miso is one of the rich essential mineral and good source of vitamin B.
I audible, sir? Am I audible? Okay. So as you can see that some of the examples for the microbiomes is the meso. Meso, if we say, it is a rich in essential minerals and a good source of the various vitamin B, vitamin E, K, and the folic acid. And the meso also provides the gut with the beneficial bacteria that help us to stay healthy, vibrant, happy. Another examples are the green peas. These are the premium and the favorable lacto-fermented peas, which we can eat as such, or even we can eat up as well with the salads also. Okay, so these are some of the examples which are used and which mainly reduce the probiotic count due to the presence of the salt and the antioxidants. So that's why we used to prefer that we will go with the miso and the green peas eating as such instead of adding the salt and oils in that. Okay, this is the some of the histories for that. As you can see that in the 1680s, Antoine Bell Leeuwenhoek, they have noted the differences in the microbes which are found in the samples from the healthy and the sick peoples. In the 1876, Robert Koch, he has established that the microorganism causes the disease. Example for that, the Bacillus anthracis that mainly cause the anthrax. Right? Okay. Another one is the 1930, where the more disease-causing microorganisms are being discovered.
Hello, India ma'am.
हाँ मैडम.
Your screen is not visible. It is shared,
Oh,
but the PPT is not visible.
okay, ma'am. I will reshare the PPT. Is it okay now, madam?
Yes, yes, madam.
Okay. And so,
What about the status of expert?
ma'am, he is getting some of the internet issues, so that's why I'm continuing with the presentation.
Okay. Otherwise, we can wait for 2 minutes.
Okay, ma'am.
Yes.
Sure, ma'am.
am audible now? Hello.
It's audible,
Hello.
sir. Yeah, yeah.
Is audible? Yes, yes, yes. Sorry,
It's
sorry.
audible, sir. I'm share
It's
the
because
presentation. You kindly start with explaining that, sir. Another 15 minutes are there, sir.
okay, okay, ma'am. Okay, okay. Thank you. Yeah, yeah. Before one. Yeah, we have understood about something about microbiome present in healthy and non-healthy conditions. Here, the uh, present we are discussing about how bacteria are healthy and unhealthy. So, bacteria present in the gut may bring about some drastic effects by mediating through causing inflammation and affecting the membrane of a cell and interacting with different receptor and entering into the cell. So they bring about harmful action also, and some of the bacteria which are how they are useful by virtue of their production, virtue of their receptor action, and then production of some acids which are very, very important for normal gut microbiota, the behavior of gut microbiota. So in this condition, we are also hearing the dysbiosis. Dysbiosis is the main reason it cause inflammation in the gut and bring about some harmful action on the wall of the stomach or wall of the small intestine or wall of the inner alimentary canal. So that we need to protect. How we're going to protect is by eating whatever I said, the healthy diet food containing one or the other two of the microbiome. Okay. Next slide, please, Bindia madam. So here we are talking about the size of the bacteria, and then they estimated that the gut of a healthy human is home to some of the 100 trillion bacteria, roughly an order of magnitude greater than the number of host somatic cells. I told about in the first slide how discussed. Number of somatic cells or vegetative cells. If you are calculating the more than that, you are going to get type of bacteria present in our healthy body, healthy system. So when you are going through a disease condition, so there will be a dysbiosis. There will be a need of modulation. That is called as immunomodulation or modulation with the help of some other type of tablet dosage form. So that in the same condition I told about diarrhea or dysentery, the person is weak because of the lack of the he or she lost the water from the body system, and how to retrieve it once again. I'm telling, stressing on, they again lose the beneficial bacteria from the gut, that is leakage through excretion. So that also we have to bring back to the normal condition. So that's why we take uh, tablet in the form of lactic acid bacteria. Okay, and then stuffed with uh, many type of like bacteroides family of bacteria or bifidobacteria in the small children if you see. So that also very plays a very important role. Okay? I think next slide I need. So new breakthroughs in the era of microbiota or microbiome research in 1950, microbiologists were able to study the host microbe relation and microbe interaction in animal through germ-free animals. So what are germ-free animals is all about. Nude mice, you heard about any bacteria which are sterile. So those are the animals which are sterile in their gut as far as possible. The gut microsystem is sterile, so that we can able to study the effect of those gut microbiome.
Any problem?
So uh, microbiome and human health, how it is connected is that, so microbiota harbor a vast community of microorganisms that are symbiotically associated with the human epithelial cells and innate immunity. So they are interconnected. They are in symbiotic procedure. So they are surviving because of the human cells also. Okay. So microbial metabolites, for instance, the lactic acid, antimicrobial peptides, and hydrogen peroxide produced by lactobacillus species in our gut keep the pH balance, maintains population of beneficial bacteria, and enhance uh, while they preventing that of the pathogens. So microbial pathogens are how to interact, how to mitigate the pathogenic bacteria is they are given here. So here is what is a thin line. Working with thin line is that an imbalance leads to we have seen imbalance leads to the dysbiosis, and which tends to decrease the population of beneficial species and increase that of pathogens, leading to the susceptibility to infectious and diseases. So we should not be susceptible to infection. In that case, we need to eat healthy diet, which is stuffed with a good type of bacteria or good type of microbiome. So our innate immune system constantly monitor the presence, the presence, uh, and so I think you're running with the short time also. Okay, because of the hindrances many times we have faced during the presentation. So microbiome and aging, how they are interconnected with the aging, how you are saying that your age of 23 and your age of 48 or age of 68, how you differ from the microbiota. If you see, I said about signature of the microbiota, if you see the gut microbiota of a healthy patient, healthy person, I'm sorry, patient, healthy person and age old person. If you see the younger person and age old person, there is difference. Healthy patient, healthy person and the non-healthy person, the microbiota is different. So if you are consuming more amount of alcohol, your gut microbiota is different. And if you are consuming good amount of probiotic food preparation like buttermilk, your microbiota is definitely beneficial in nature. So small babies which are born, they are sterile in their womb, and they are born exposed to the environment. They will colonize with one other the type of bacteria in start the day one of their birth. So this is how you are going to monitor is that how microbial come to rescue is that microbial interaction that the pathogenic bacteria take over the microenvironment in the host babies and causing distress or dysbiosis. The antimicrobial activity of beneficial bacteria in the microflora help uh, to rectify this infection by killing the pathogens. And microbial infections are the range from acne to sexual transmitted diseases, HIV. Antimicrobial activities are now being explored, and many type of therapeutic uh, form of tablet they came with the help of probiotic. They are treating many diseases, and they are also in formulations they are giving. So next slide, please. So chronic diseases and immunomodulation. I told about immunomodulation. That the preparing our body cells or body system to fight with the disease. That is what an active immunity is all about. And this also we can able to bring to the normal condition with the help of uh, bacteria or microbiome stuffed with food and both as a medicine. We can able to maintain the balance of the microbiota in the food, and we can able to overcome the chronic conditions also. So there are many reports are there where cancer is also because of the dysbiosis. In cause of the cancer is many time dysbiosis, and gut brain axis is all about the area where nowadays people are focusing, and how our gut bacteria is influencing the brain. There are many this have been studied. For example is multiple sclerosis. Next slide, please. Recent advancements in the field of microbiota studies are few are listed here. One is from Nature Biotechnology. and metagenomic systems biology, where gut microbiota reveal the association of body system and the disease condition. Next slide, please. Manipulating. What is manipulating? The adding a particular type of bacteria through dosage form, through tablet form, through some of the exercises. Okay, so manipulating the gut microbiota or method and challenges are many. So we are hearing deficiencies of many of the type. We are using here animal models that to demonstrate germ-free animal models have been utilized to study the effect of microbiota on metabolism. So that is also studied through germ-free animals. Next slide. So these are some of the papers which tell about how the probiotic things or probiotic bacteria, probiotics are very, very important in maintaining the health and disease, how to overcome, mitigate the disease condition in the individual. Next slide. These are some of the reports or references where we have taken that tells about importance of gut microbiota. Next slide, please, ma'am. So what are the challenges of present thing is many of the species and strains are non-culturable, meaning they do not grow in lab conditions. That is a challenge that we need to find it out, and many isolation techniques should be improved to identify particular type of bacteria, fungus, and viruses, and to develop the media required for their growth and metabolism. That is very, very important. That is a challenge here that we need to grow. And sci-fi or future trend, what is the future trend? Artificial intelligence and synthetic biology approaches for human gut microbiota is also being monitored, has been used nowadays to understand the gut microbiota in a systematic manner and how they are connected with the aging, especially that we need to identify through artificial intelligence. So these are some of the references what we have referred, what I referred, and my team. I thank Shivani Tendulkar and Ashwarya Hattivali for preparing the slide, and earlier we have discussed about this, and my colleagues. I told about Dr. Ramesh Paranjaped, Abhijit Bhatkal, Bhushan Kulkarni, Deshpande, Ritya Upin, and Dhanashri is my colleagues. I'm thank for entire team for whatever I got knowledge from last few years. Thank you for the opportunity.
Hello, sir.
Good evening, ma'am.
Yeah, good evening, sir. So, thank you so much for giving your precious time. And as a token of appreciation from the family of the Parul University, we would like to facilitate you with an e-certificate. So, sir, with your permission, I would like to share my screen, sir.
Okay, ma'am.
Sir, kindly accept this e-certificate from our side.
Thank you for this opportunity and encouragement from your side. I'm very sorry for technical issues from my side, but it was entirely the new
It
opportunity
happens,
for me.
sir. It
Yeah,
happens. Sir. If the online sessions are there, sometimes the technical issues happen which are not in our hands, sir. But still, we are thankful to you, sir. Ki within the time frame you have completed the our this complete 1 hour webinar, sir.
yeah. I thank my entire team. I think.
No, thank you so much, sir, for enlightening us on such an informative presentation, and it was a very informative session, sir.
Yes, sir. Thank you,
Thank
sir.
you so much for the opportunity and time and dedication from your side. Thank you.
Yes, sir.
Yes, sir. It was very nice session. Thank you.
Thank you, ma'am.
Yes, sir. And suppose you are coming to Baroda, please visit Parul University.
Definitely, ma'am, it's an opportunity for me. My one of the teacher is already serving in Port University, Dr. Girish Babu. He's presently HOD of I think I believe Life Science Department.
Yes, very nice. So we
Yes,
can
thank you
even
so
meet
much.
that here in
Yes, ma'am.
university also. Thank you.
Yes, ma'am. Yes. Thank you so much, ma'am.
Thank you so much, sir.
Thank you, ma'am.
If anybody having any question or the doubt, so the session is open for the question and answer.
I think, madam, in the next time, whenever I get an opportunity,
No
I
issue,
think
sir.
we will discuss some other aspects of microbiota, what we are working presently in our department.
Sure, sir. And even after the session, also, if anybody got any question and doubt, he kindly send me the all the question, and I will ask to the sir to answer it later. And I have shared the feedback form link in the chat box, so everyone kindly fill that.
Yes, ma'am. Yes, ma'am.
Yeah, all the participants are requested to fill the feedback form for this session. If anybody is not having the question, so here we are concluding this the webinar. Thank you, sir.
Thank you, ma'am. Thank
Goodbye,
you for the
sir.
opportunity. Yes, thank you.
Okay, thank you, everyone. Have a good day.